The analytical methods performed to assess quality of fishery products. Quality refers to the aesthetic appearance and freshness or degree of spoilage which the fish has undergone. It may also involve safety aspects such as being free from harmful bacteria, parasites, or chemicals. It is important to remember that quality implies different things to different people and is a term which must be defined in association with an individual product type. The methods for evaluation assessing fish or fish product quality may be conveniently divided into two categories: the sensory and instrumental. Since the consumer is the ultimate judge of quality, most chemical or instrumental methods must be correlated with sensory evaluation before being used in the laboratory. However, sensory methods must be performed scientifically under carefully controlled conditions so that the effects of test environment, personal bias, may be reduced. First, we have sensory methods, defined as the scientific discipline used to evoke, measure, analyze, and interpret reactions to characteristics of food as perceived through the senses of sight, smell, taste, touch, and hearing. Most sensory characteristics can only be measured meaningfully by humans. However, advances are being made in the development of instruments that can measure individual quality changes. So this is Instron Ballon Rheometer. This is an instrument capable of measuring parameters included in the sensory profile. Ballon rheometer is for measuring texture and other rheologic properties. Microscopic methods combined with image analysis are used to assess structural changes, and the artificial nose to evaluate odor profile. Sense the sensory process. In sensory analysis, appearance, odor, flavor, and texture are evaluated using human senses. Scientifically, the process can be divided into three steps. First, the detection of a stimulus by the human sense organs. Second, evaluation interpretation by a mental process. And third, the response of the assessor to the stimuli. Variations among individuals in the response to the same level of stimuli can vary and can contribute to an unconclusive answer of the test. People can, for instance, differ widely in their response to color and also in their sensitivity to chemical stimuli. Some people cannot taste rancid flavor, and some have a very low response to cold storage flavor. Assessment of fishery products can both be performed as a discriminative test and as a descriptive test. First, we have discriminative test. In discriminative test, we have a triangle test. So this is the most used discriminative test in sensory analysis of fish is the triangle test, which indicates whether or not a detectable difference exists between two samples. The assessors receive three coded samples, are told that two of the samples are identical and one is different, and are asked to identify the odd sample. Analysis of results from the triangle test is done by comparing the number of correct identifications with the number you would expect to obtain by chance alone. In order to test this, the statistical chart in Appendix A must be consulted. The number of correct identifications is compared to the number expected by use of a statistical table. If the number of responses is 12, there must be 9 correct responses to achieve a significant answer. Triangle tests are useful in determining if ingredient substitution gives a detectable difference in a product. Triangle tests are often used when selecting assessors to a taste panel. So these as the samples marked A and B can be presented in 6 different ways. So the equal numbers of the 6 possible combinations are prepared and served to the panel members. They must be served randomly, preferably as duplicates. The number of panel members should be no less than 12. So this is an example of score sheet triangle test. Here we have name, type of sample, the date, test sample number, describe the difference, and the two of these three samples are identical, the third is different. So this is to examine the samples from left to right and circle the number of the test sample which is different. It is also essential you make a choice and guess if no difference is apparent. We have the ranking. In a ranking exercise, a number of samples are presented to the taste panel. Their task is to arrange them in order according to the degree to which they exhibit some specified characteristics, downward concentration of salt. Usually, ranking can be done more quickly and with less training than evaluation by other methods. Thus, ranking is often used for preliminary screening. The method gives no individual differences among samples and is not suited for sessions where many criteria have to be judged simultaneously. Profiling. Descriptive testing can be very simple and used for assessment of a single attribute of texture, flavor, and appearance. Methods of descriptive analysis, which can be used to generate a complete description of the fish product, have also been developed. An excellent way of describing a product can be done by using flavor profiling. The quantitative descriptive analysis provides a detailed description of all flavor characteristics in a qualitative and quantitative way. The method can also be used for texture. The panel members are handed a broad selection of reference samples and use the samples for creating a terminology that describes the product. In Ling, in Lingbai, a descriptive sensory analysis for fish oil using quantitative descriptive analysis has been developed. A trained A trained panel of 16 judges is used. Descriptive terms such as paint, nutty, grassy, metallic are used for describing the oil on an intensity scale. A moderately oxidized fish oil is given fixed scores and used as a preference. So this is the profile of fish oil. We have here the taste, the fresh if it is fresh fish, the amin, or if it is oily, sweet, metallic, grassy, painty, fruity, and the remarks and taste as a whole. Advanced multivariate analysis is used for statistics and makes it possible to correlate single attributes to oxidative deterioration in the fish oil. The results can be reported in a spider web. The panel uses an intensity scale normally ranging from 0 to 9. Profiling can be used for all kinds of fishery products, even for fresh fish, when special attention is placed on a single attribute. The results of quantitative descriptive analysis can be analyzed statistically using analysis of variance or multivariate. We have statistics. In any experiment including sensory analysis, the experimental design of panel members, number of samples, time aspects, hypothesis, and statistical principles should be planned beforehand. Failure to do so may often lead to insufficient data and non-conclusive experiments. A guide to the most used statistical methods can be seen in Mailguard, 1991. A panel used for descriptive testing shall preferably consist of no less than 8 to 10 persons, and it should be remembered that the test becomes statistically much stronger if it is done in duplicate. This can often be difficult using sensory analysis on small fish. Thus, the experiment must include a sufficient number of samples to remove the sources of variability, and the testing must be properly randomized. For further information, we have flavor profiles. This is the flavor profiles of a fish oil after 2 weeks of storage at various temperatures according to Rohrbeck 1993 and others. Training of assessors. The training of assessors for sensory evaluation is necessary in almost all sensory methods. The degree of training depends on the difficulty and the complexity of the assessment. For example, for profiling, a thorough training with presentation of a large range of samples is necessary in order to obtain proper definitions of the descriptors and equal use of the scoring system. The triangle test normally requires a minor degree of training. Sensory quality control is often done by a few persons either at the fish market when buying fish or at quality inspection. The experience of these persons allow them to grade the fish. Starting as a fish inspector, it is not necessary to know all the different methods of sensory assessment described in these textbooks, but some of the basic principles must be known. The assessor must be trained in basic tastes, the most common fish taste, and must learn the difference between off, flavor, and taints. This knowledge can be provided in a 2-day basic training course. In bigger companies and for experimental work, a further training of a sensory panel is necessary in order to have an objective panel. A laboratory panel must have 8 to 10 members, and the training and testing of panel members must be repeated regularly. Next, we have the facilities. The minimum requirement for evaluation is a preparation room and a room where the samples are served. The rooms should be well ventilated and provided with a good light. There must be enough space on the tables for inspection of raw samples of fish. Lastly, we have the cooking and serving. The samples of fishery products should not be less than 50 to 100 grams per person. Fillets can be served in loins and should be cooked on internal temperature of 65 degrees Celsius. The samples should be kept warm and served in insulated containers or on a hot plate. The fish can be heat treated by steaming in a water bath, packed as boiled in the bag in a plastic pouch or in a alufoil. An oven can also be used for heat treatment. The fish can be packed in plastic or put on a small porcelain plate covered with alu foil. For cod loins on a porcelain plate covered with alu foil, the heating time in a steam oven at 100 degrees Celsius must be 10 minutes. The sample should be coated before serving. Next, we have the biochemical and chemical methods. The appeal of biochemical and chemical methods for the evaluation of seafood quality is related to the ability to set quantitative standards. The establishment of tolerance levels of chemical spoilage indicators would eliminate the need to base decisions regarding product quality on personal opinions. Of course, in most cases, sensory methods are useful for identifying products of very good or poor quality. Thus, biochemical or chemical methods may best be used in resolving issues regarding products of marginal quality. In addition, the biochemical or chemical indicators have been used to replace more time-consuming microbiological methods. Such objective methods should, however, correlate with sensory quality evaluations, and the chemical compound to be measured should increase or decrease with the level of microbial spoilage or autolysis. It is also important that the compounds to be measured must not be affected by processing. So this is the overview of some of the most useful procedures for the objective measurement of seafood quality. So Woyewoda and others in 1986 have produced a comprehensive manual of procedures. This includes the approximate composition of seafood. First, we have the amines, or the total volatile basic amines, one of the most widely used measurements of seafood quality. It is a general term which includes the measurement of trimethylamine produced by spoilage bacteria, dimethylamine produced by autolytic enzymes during frozen storage, and ammonia that is produced by the deamination of amino acids and nucleotide catabolites, and other volatile basic nitrogenous compounds associated with seafood spoilage. Next, we have ammonia. Ammonia is formed by the bacterial degradation, deamination of proteins, peptides, and amino acids. It is also produced in the autolytic breakdown of adenosine monophosphate in chilled seafood products. Although ammonia has been identified as a volatile component in a variety of spoiling fish, few studies have actually reported a quantification of this compound, since it was impossible to determine its relative contribution to the overall increase in total volatile bases. Trimethylamine. Lianin. The trimethylamine is a pungent volatile amine often associated with a typical fish odor of spoiling seafood. Its presence in spoiling fish is due to the bacterial reduction of trimethylamine oxide, which is naturally present in the living tissues of many marine fishes. Although it is believed to be generated by the action of a spoilage bacteria, the correlation with bacterial numbers is often not very good. This phenomena is now thought due, to be due to the presence of small numbers of specific spoilage, bacteria which do not always represent a large proportion of the total bacteria flora, which are capable of producing large amounts of spoilage-related compounds such as the trimethylamine. One of these specific spoilage organisms, Protobacterium phosphorium, generates approximately 10 to 100-fold the amount of trimethylamine than that produced from the more commonly known specific spoiler, Shewala putrefaciens. Dimethylamine, that is produced autolytically during frozen storage. For gadoid fish such as hake, it has been found to be a reliable indicator of Fa-induced toughening because it is associated with membranes in the muscle. Its production is enhanced with rough handling and with temperature fluctuations in the cold storage facility. Dimethylamine has little or no effect on the flavor or texture of the fish per se. but is an indirect indicator of protein denaturation, which is often traceable to improper handling before and or during frozen stage. Next, we have biogenetic amines. Fish muscle has the ability to support the bacterial formation of a wide variety of amine compounds, which result from the direct decarboxylation of amino acids. Most spoilage bacteria possessing decarboxylase activity to do so in response to acidic potential hydrogen. Presumably that the organisms may raise the potential hydrogen of the growth medium through the production of amines. So histamine, putrescine, cadaverine, and tyramine are produced from the decarboxylation of histidine, ornithine, lysine, and tyrosine respectively. Histamine will receive most of the attention since it has been associated with the incidence of scombroid poisoning in the conjunction when the ingestion of tuna, mackerel, mahi-mahi. So, however, the absence of histamine in scombroid fish does not ensure the wholesomeness of the product since spoilage at chill storage temperatures does not always result in the production of histamine. So, this table is Smith's and Karma's 1977 proposed a chemical quality index based on biogenetic amines which reflected the quality loss in canned tuna. So this is the table where quality index is equals to ppm histamine plus ppm putrescine plus ppm cadaverine is divided into 1 plus pm spermidine plus ppm spermine. Next, we have ethanol. Ethanol has been used for many years as an objective indicator for seafood quality, although it is not nearly as common as the analysis of trimethylamine. Since ethanol can be derived from carbohydrates via anaerobic fermentation and deamination and decarboxylation of amino acids such as alanine, it is a common metabolite of a variety of bacteria. It has been used to objectively measure the quality of a variety of fish, including canned tuna, canned salmon, raw tuna, and redfish, pollock, flounder, and cod. To date, the simplest and perhaps most reliable means of measuring ethanol in fish tissue is the use of the commercial enzyme test kits available from Boehringer, Manim, or diagnostic chemicals. One advantage of using ethanol as a spoilage indicator is that it is heat stable and may be used to assess the quality of canned fish products. Next, we have the third method is the physical methods. In physical methods, we have electrical properties. It has long been known that the electrical properties of skin and tissue change after death, and this has been expected to provide a means of measuring post-mortem changes or degree of spoilage. However, many difficulties have been encountered in developing an instrument. For example, species variation, within a batch of fish, different instrument readings when fish are damaged, frozen, filleted, bled, or not bled, and a poor correlations between instrument reading and sensory analysis. Most of these problems, it is claimed, are overcome by the GR thermometer. However, the instrument is not able to measure quality or freshness of a single fish, although it may find application in grading batches of fish. So this is the relation. This graph is the relationship between GR thermometers readings of various species of fish and freshness adapted from Chain 1975. So until recently, no instruments have been capable of an online determination of quality, although this type of mechanized quality evaluation would be highly desirable on the processing floor. The RT freshness grader development began in 1982, and by 1990, a production model capable of sorting 70 fish per minute over 4 channels was made available. The developer was Rafagnaticny Electronics, based the sensing unit on the GR thermometer. Next, we have the potential hydrogen and the oxidation reduction. Knowledge about the potential hydrogen of fish meat may give valuable information about its condition. Measurements are carried out with a potential hydrogen meter by placing the electrodes either directly into the flesh or into a suspension of fish flesh in distilled water. Measurements of oxidation reduction are not carried out routinely, but it is likely that a freshness test can be based on this principle. Next, we have measuring texture. Texture is an extremely important property of fish muscle, whether raw or cooked. Fish muscle may become tough as a result of frozen storage or soft and mushy as a result of autolytic degradation. Texture may be monitored organoleptically, but there has for many years been a need for the development of a reliable objective rheological test which would accurately reflect the subjective evaluation of a well-trained panel of judges. So, Gill and others in 1979 developed a method for evaluating the formaldehyde-induced toughening of frozen fish muscle. The method utilized an Instron model TM equipped with a Kramer shear cell with 4 cutting blades. This method correlated well with the data obtained from a trained texture panel, a method for measuring hardness, soft of fish flesh, designated as compressive deformability, has been reported by Johnson and others in 1980. All accurately cut fish sample is compressed by a plunger and a stress strain curve recorded. A modulus of deformability is calculated from the recorded graph. The results from such measurements may, however, be difficult to interpret. Another method measuring the shear force of fish flesh has been investigated by Dunajski, 1980. From this work, it has been concluded that a thin-bladed shear force cell of the Kramer type can be applied. So the fourth method is the microbiological methods. The aim of microbiological examinations of fish product is to evaluate the possible presence of bacteria or organisms of public health significance and to give an impression of the hygienic quality of the fish, including temperature abuse and hygiene during handling and processing. Traditional bacteriological examinations are laborious, time-consuming, costly, and require skill in execution and interpretation of the results. It is recommended that such analysis can be limited in number and extent. Various rapid microbiological methods have been developed during the last decade, and some of these automated procedures may be of use when large numbers of samples are to be analyzed. So, in microbiological methods, we have first the total counts. This parameter is synonymous with total aerobic count and standard plate count. The total count represents, if carried out by traditional methods, the total number of bacteria that are capable of forming visible colonies on a culture media at a given temperature. This figure is seldom a good indicator of the sensory quality or expected shelf life of the product. In ice-stored Nile perch, the total count was 109 colony forming unit per grams for days before the fish was rejected. And in lightly preserved fish products, high counts prevail for a long time before rejection. If a count is made after systematic sampling and a thorough knowledge of the handling of the fish before sampling, temperature conditions, packaging, it may give a comparative measure of the overall degree of bacterial contamination and the hygiene applied. However, it should also be noted that there is no correlation between the total count and presence of any bacteria of public health significance. So the common plate count agars are still the substrates most widely used for determination of total counts. However, when examining several types of seafood, a more nutrient-rich agar gives significantly higher counts than PCA. So furthermore, the iron agar yields also the number of hydrogen sulfide producing bacteria, which in some fish products are the specific spoilage bacteria. Incubation temperature at and above 30 degrees Celsius are inappropriate when examining seafood products held at chill temperatures. Poor plating and a 3 to 4 day incubation at 25 degrees Celsius is relevant when examining products where psychotropha Psychotrophs are the most important organisms, whereas products where the psychrophilic Photobacterium posperium occurs should be examined by surface plating and incubation at maximum 15 degrees Celsius. So this table is the methods for determination of the content of bacteria in seafood. We have the method, the temperature in degrees Celsius, the incubation, and the sensitivity or um, colony forming unit per grams. Spoilage bacteria. So the total number of spoilage bacteria on fish rarely indicates sensory quality or expected storage characteristics. However, it is recognized that certain bacteria are the main cause of spoilage. Different peptone-rich substrates containing ferric citrate have been used for detection of H2S producing bacteria such as Shewanellia putrefaciens, which can be seen as black holiness due to precipitation of um, FeS or sulfate. sulfide. Ambient spoilage is often caused by members of Vibroniaceae that also will form black colonies on an iron agar to which an organic sulfur source is added. No selective or indicative medium exists for the Pseudomonas that spoil some tropical and freshwater fish or for Photobacterium phosphorium that spoiled packed fresh fish. So the presence or absence of pathogenic bacteria is often evaluated by methods based on immuno or molecular biology techniques. Such techniques may also be developed for a specific spoilage bacteria, and the technological laboratory has been currently investigating the use of antibodies specific for S. putrefaciens. So, spoilage reactions. Several spoilage reactions can be used for evaluation of the bacteriological status of fish products. As described above, agars on which H2S producing organisms are counted have been developed. During spoilage of white plain fish, one of the major spoilage reactions is the bacteriological reduction of trimethylamine oxide to trimethylamine. When trimethylamine oxide is reduced to trimethylamine, several physical changes occur. The redox potential decreases, the potential hydrogen increases, and the electrical conductance increases. The measurement of such changes in a trimethylamine oxide containing substrate inoculated with the sample can be used to evaluate the level of organisms with spoilage potential. Thus, the more rapid the change occurs, the higher of the level of spoilage organisms. So that's the end of the slideshow, and thank